In this video, we'll be looking at the targets of drugs. And here are the learning outcomes that we'll be looking at in this video. I'd like to start thinking about how cells communicate. And so we've got two white blood cells here. And one thing you won't hear them discussing is the bugs are invading and deciding to go and get them. And the reason why is because they don't have eyes and ears and mouth and nose to actually have this conversation. So how exactly does a white blood cell know that there are invading bacteria and another blood cell know that maybe they should join the foray and take care of the invading bacteria? And this goes with any type of event or scenario that our bodies encounter. Cells need to communicate with each other. And so the tool that is used by cells to communicate with each other is known as a receptor. Now, this is usually a macromolecule, and it's usually a protein. These proteins are able to bind to endogenous molecules, and these endogenous molecules are the signaling molecules that go from one cell to another, or from one part of the body to another part of the body. Now, when we start thinking about drugs, is that they usually mimic an endogenous molecule. So we're not kind of reinventing the wheel. What we're doing is piggybacking on a whole lot of homeostatic mechanisms and systems that are already in our bodies and already functioning. And we're just going to modify them. And we do that by using drugs that look like endogenous molecules. With receptors and drugs, when they interact with each other, because they will physically interact with each other, there is a change in the shape and function of the receptor. And so it's a way of changing the way a cell behaves when we use a drug. Now, the receptor signal doesn't stop at that protein. It actually initiates a chain reaction of secondary messengers that propagate that message and continue that communication through the cell to the propagators or the actual mode of action that is actually going to make the change. And these receptors generally have a normal role in physiological or pathophysiological conditions. So just a couple of examples here is we have acetylcholine and nicotinic receptors. And that's involved in skeletal muscle contraction. And we also have adrenaline and beta-2 adrenoceptors, and they're involved in airway smooth muscle relaxation. What we have here is just a list of some of the main protein families of drug targets. First one on the list is the G protein coupled receptor. The second is nuclear hormone receptors, the ion channel family, enzymes and kinases, which are really part of enzymes as well, and transporters. And another one which is not as frequently seen is the catalytic receptors. So the first one on the list is the G protein coupled receptor. Now, this protein is the G protein coupled receptor. And down the bottom is the G protein itself. And it's made up of the alpha and a beta gamma subunit. And the beta gamma subunit, or the whole G protein, is actually on the intracellular surface. Whereas the G protein coupled receptor sits across and spans the membrane. So when a ligand or endogenous ligand or a drug interacts with a G protein coupled receptor, it actually sits on the surface or on that extracellular aspect of the G protein coupled receptor. And what it does is it causes a change in the G protein coupled receptor, which affects the G protein itself, and the G protein decouples and moves. Now, the reason why the G protein is called a G protein is because it couples with GTP, and there is exchange of GDP and GDP at this point, and that determines how long the alpha subunit actually does its functioning parts. Now, just to point out that the beta gamma subunit is also functional. Now, some of the things or other proteins that G proteins interact with are kinases, so protein kinase A and C are example, cyclases like adenylate cyclase, but they can also interact with ion channels like the G protein coupled inward rectifying potassium channels, so they can also change membrane potential. Now, these are fairly quick onset, so their time to effect is when the ligand binds, it's going to be anywhere between seconds to hours, depending on what secondary messenger pathways are affected. So it's a fairly quick onset. So some examples that you might see that are G protein coupled receptor are the adrenaline receptors or adrenergic receptors, dopamine receptors, or the enkephalin receptors that respond to morphine. The next family is the nuclear hormone receptor. Now, this time, the receptor is actually inside the cell completely. And so it's found either in the cytoplasm or in the nuclear kind of area. Now, the ligand itself, this time, has to get through the plasma membrane to interact with the nuclear receptor. Now, the nuclear receptor itself dimerizes, so there's usually two parts of it, so one part over here and one part over here. Sometimes they're the same, so a homodimer forms, or it could be a heterodimer forming, so they're actually two different nuclear hormone receptors that bind together, and it's the dimer that is the active form. Now, once the ligand has bound, or both ligands are bound, then that moves into the nuclear area where it interacts with DNA and changes transcription. So this is actually a slower onset of action. So the time to effect is hours to days. So any drug that uses this mechanism is going to take time to actually work. So some examples here are the estrogen receptors, thyroid receptors, and glucocorticoid receptors. These are all nuclear hormone receptors. So the next family that we're going to be looking at are the ion channels. Now, these can be activated by a ligand, or they can be changed or activated by a membrane potential change. And there are two sets of family. So in this case, we're looking at a ligand gated one. So here's the ligand, drops onto the ion channel, and it opens up and allows electrolytes to flow through that channel. In this case, we're looking at sodium moving into the cell. And what this does is it causes a change in the membrane potential. So neurons use this, muscles use this to enact action potentials or to enact contraction. Now, this is a very rapid onset of action. So the time to effect, you're looking at milliseconds to seconds to see a change in the behavior of the cell. And the examples I'll provide here are the acetylcholine receptor, in particular the nicotinic receptor. So this is an ion channel. The GABA aminobutyric acid, or GABA receptors. These are chloride channels. And there's also the voltage-gated sodium channels, which will change when there's membrane potential change, or become open when there's membrane potential change. So this family we're going to look at are the enzymes. Now, they can be found in lots of different places. They can be soluble in the blood, they can be attached to membranes on the surface of cells, or they can be on intracellular aspects where they might be on a surface of an organelle inside the cell. Now, the time to onset is going to be variable depending on what the enzyme is doing and whether it's the substrate that is inactive or the product. So just a brief review of the enzymes and how they're working is that the substrate will bind, there will be a change in the conformational state of the enzyme itself, and you'll end up with a product. So the substrate will have its own impact and the product will have its own impact. And so a few examples here is we have the angiotensin converting enzyme, which is used in hypertension, dipeptidyl peptidase, number 4, which is used in diabetes, and phosphodiesterase is another. This one's an intracellular enzyme which is used in or targeted in things like asthma or stroke or pulmonary hypertension is another example that you might see these agents used. So although kinases are a part of the enzyme family, we're going to focus on just one aspect of the kinases because they seem to be the main drug targets. So the example here that we're going to be looking at are the tyrosine kinase receptors. So the tyrosine kinase receptors are like many of the other integral membrane protein receptors in that they have an extracellular component, which is where the ligand binds. There is a transmembrane domain region, and then there is the intracellular region. Now, when the ligand binds, the receptor dimerizes. And so when the receptor dimerizes, it creates autophosphorylation of the receptor. And so phosphate in this case is acting like a molecular switch, and the kinase is adding on phosphates to the tyrosine residues or amino acids that are on that intracellular component. And this changes the way that that intracellular domain reacts with other proteins around it. And so the pathways that get activated is a series or a cascade of kinases. And so the tyrosine kinase receptor will also phosphorylate this other kinase, which will then dissociate and then go and activate other kinases within inside the cell. And it will do this to a number of other kinases, and eventually the cell will change its behavior. Now, the time to effect is actually fairly rapid when you consider the number of steps. But it does start within minutes and has effects out to kind of days. Now, the example here is the insulin receptor, which regulates how glucose is controlled. Also the VEGF, or vascular endothelial growth factor receptor family, and the ERB, or endothelial growth factor family, receptor family, are examples of tyrosine kinase receptors. So the transporters is the last family that I would like to talk about in this video. And the role of the transporter is to move the substrates from one side of the membrane to the other side of the membrane. And usually there are two main mechanisms that these transporters work by. One is to use a sodium gradient, and because sodium is always wanting to get into the cell, it's useful for pulling substrates into cells. If you're working against a concentration gradient, then maybe ATP may be required to initiate or provide the energy for that transport. In this example here, I've got sodium and I've got serotonin. So this is a serotonin transporter. What will happen is that will go together through the transporter, and it will facilitate the movement of serotonin into the cell. Now, the time to effect is really dependent on the substrate and its effects on tissues. So the couple of different examples here is the serotonin transporter, which is a target of drugs that are used for treating depression and anxiety. Another example which uses ATP as a driving force is the hydrogen potassium ATPase, which is found in the stomach, which provides the protons for acidification of the stomach contents. And also the sodium glucose transporter, which is used for moving glucose, absorbing it or reabsorbing it in the kidneys. In the next few slides, I just want to point out that there are some other drug targets other than the classical protein targets we've just discussed. So DNA and mRNA can both be drug targets. So for example, the intercalating agent doxorubicin binds in between nucleotide pairs in DNA chains, and this disrupts topoisomerase, which is an enzyme that winds and unwinds or coils DNA. There is also antisense forms of drugs, so nusinersen, which is a drug that binds to mRNA and alters the splicing mechanism and expression of the survival motor neuron gene, and used for spinal muscular atrophy. Then we have gene transfer, where the example here is voretigene, which is a viral delivery of a new gene or a replacement gene of RPE65. So this is a retinal pigment epithelial protein, which is 65 kilodaltons in size, and it's an isomerase, so it flips vitamin A from its inactive form to its active form. You can also have osmotic type drugs, which don't interact with any proteins or mRNA or DNA, they interact with water. And so diuretics like mannitol draw water to themselves and therefore increase urine volume. There are also laxatives that use this same mechanism. So for example, sorbitol, which draws water to it, softening the stool. Then we can look at those that are chemical reaction mediated. So for example, antacids. So for example, magnesium hydroxide reacts with hydrochloric acid in the gastric lumen and neutralizes it. Or you can have drugs that bind to important targets. In this case, sodium edotate is binding to lead, so it's used to detoxify somebody who's had a lead exposure. Or, and this is where a lot of drugs seem to be going, is antibody binding. So a monoclonal antibody is used to bind to an important biomolecule of the body. In this case, adalimumab is binding to tumor necrosis factor alpha, and that alters the way the immune system functions, particularly useful in something like rheumatoid arthritis, where you've got an autoimmune condition. So as far as learning outcomes, hopefully you should be able to describe the general features and the function of the receptor. So how does it work? What does it do? What are the kind of pathways that could be initiated via that receptor? Should be able to identify at least 6 common drug targets and to differentiate those cellular pathways they use, where they're found, and how long they take to actually affect a change to cell function.